Welcome to Pharmacognosy and Metabolic Engineering, one of the NPTEL online certification course. So, we will go to lecture number 29. So, where I am going to show today about the cell and tissue localization of terpenoid indole alkaloids. So, I will do some drawing in the board and the enthusiastic students may also draw along with me in the board. So, that their concept will get clear. Let us go to the board. So, what I am going to cover here is the compartmentation of terpenoid indole alkaloid pathway in Catharanthus roseus. Okay. So, what we have seen that the different enzymatic states are operating within the cell. So, let us make a heading for this cellular localization of both enzymes and intermediates during terpenoid indole alkaloid biosynthesis. This is a cell, okay, and I will put the vacuole here, maybe a slightly and this is the chloroplast. We will start with say in the chloroplast MEP pathway is operating, which color I will use, that leads to the formation of geraniol, and this geraniol will be transported out and moves into the vacuole, whereas the another source of geraniol will be from the cytosol, where MVA pathway may also contribute to the formation of geraniol. So, this geraniol will be converted into 10 hydroxygeraniol by the enzyme geraniol 10 hydroxylase, which I will put in brown color. and eventually from this geraniol second will be synthesized, and where tryptamine is also present, and this leads to the formation of strictocidin. Tryptamine comes from tryptophan. So, this tryptophan may also come from plastid localized Sekimate pathway. So, here what I will do, I will do slight changes in the drawing. So, this is tryptophan, and this geraniol is coming from MEP, that is also operated inside the plastid. I think now it is clear to all of you, and this strictocidin, one it is produced inside the vacuole, this will go out into the cytosol. So, how it is passing through the cytosol is again an interesting question. Are there any transporter responsible for that? So, this I will answer in one of the subsequent classes. So, I will not talk about this today. So, this strictocidin will be now out, and it will be subsequently converted into strictocidin aglycone by the action of SGD. So, SGD we must locate SGD. Okay. So, the arrow to be made further, okay. And then this leads to the formation of 421 gaseocyzidin. So, I will note the full form here because of the shortage of space. Gaso, and then from there one route goes towards tebarsonin, and other route produces catharanthine. This catharanthine will again enter into the vacuole, and this will be the tebarsonin. This tebarsonin now moves into the through cytosol and moves through the cytosol into the chloroplast. So, if I put the arrow, right. So, here we have the endoplasmic reticulum ER, where the T16H is localized. So, the pathway moves from traversin, and then from here it moves through cytosol. And then it enters into the chloroplast, and then a reaction catalyzed by this will be NMT, and then the next step D-acetyl vindoline will be there. Oh, so the arrow has to be, the color of the arrow has to be changed. Sorry, this is taking time, but if you draw with me, then you can visualize the pathway. And then it makes vindoline, and this vindoline again enters into the vacuole. So, catharanthine enters, vindoline enters, and these form vindoline. This joins together by a peroxidase and form the vinblastine. Well, so the enzyme responsible for this is peroxidase, and here lies the DAT, the D4H is here. Okay. And another route from this also makes the cathenamine, and that makes azmalicin, and azmalicin enters into the vacuole and converted into serpentine. So, now let us define. This is basically the chloroplast, this is the cytoplasm, this is the endoplasmic reticulum, and this one is the vacuole. So, this is in brief the cellular localization of both enzymes and intermediates during terpenoid indole alkaloid biosynthesis. So, once this is clear, now we will see another drawing to from the anatomy point of view. Let us go to the next slide. So, here basically I will write putative trafficking of pathway intermediates and products in monoterpenoid indole alkaloid metabolism. So, here I will draw a structure transverse section of the leaf. Say on the top of it is a cuticle layer, and then next to the cuticle is basically the epidermis. So, epidermis consists of epidermal cells then you have the palisade parenchyma, and then one of these you will have the palisade idioblast, and next you have this. And also you have another specialized okay. Then I think I will use this color is better. You have spongy cells, so these are the vascular tissue. So, these are IPAP IPAP stands for internal phloem associated parenchyma. Okay. So, this is sorry. Well, now let me define palisade idioblast. This is the palisade idioblast, and this is the spongy mesophyll idioblast. This is called latissifer. Okay. Now, let us see what is there. So, first of all, this is cuticle. So, this is the epidermis. So, now here we will see the pathway. The tryptophan makes tryptamine, joins with secologanin, makes streptocidin. Up to from there so we write 16-methoxy tabarsonin, then this moves so this is MIA intermediates. This further moves down to here, this also goes down to here. So, this is palisade idioblast where diacetyl indole converted to indole by the this one. Similarly, here also same thing happens, the diacetyl indole converted into indole. And here the MAP pathway originates, it makes geraniol, and that subsequently moves and this also moves to here also, where the acetyl vindoline converted into vindoline. Okay. So, this is palisade parenchyma. So, what we see that in the leaf structure, normally leaf contains the palisade and spongy parenchyma, but here what you see that there are specialized structures formed. One is called latticephore, another is called idioblast. So, these latticephore and idioblast, these are specialized cells. So, this contains latex, and why it contains latex? Because the late steps of dimeric indole alkaloid or the monoterpene indole alkaloids are toxic to the cell itself. Therefore, diacetyl vindoline and maybe the subsequent vincristine, anhydro vinblastine, these are toxic to the cells. So, plant has developed specialized cells to accommodate these toxic metabolites, and these specialized cells are latticephore and idioblast, which contains latex, and these metabolites these monomeric and dimeric indole alkaloids are stored in the latex, so that it is safe for the plant species as well. So, that is why this is. So, both in the palisade as well as the spongy parenchyma, the idioblast are there. So, this is in brief the this is basically a transverse section of the leaf. So, if you ask me to put a caption for this, so this figure is basically the transverse section of a Catharanthus roseus leaf portion. So, this is in brief the anatomical structure of the leaf tissue and where the pathway operates, but still it is not complete, because I just I will make before I end this class, I will make a short list here. So, this latticephore and the idioblast, as I said. So, here this portion D4H, DAT, these enzymes are localized, whereas in the leaf epidermis all other enzymes are localized, such as TDC, STR, SGD, T16H, OM2, not only even T3 oxidase, T3 reductase and NMT. So, the epidermal tissues are very active tissue for the indole alkaloid biosynthesis. Now, I may end the class here, but in the next class I will show you another pictorial diagram, where I will show that again this structure, but there what I will put the mesophyll tissue, phloem parenchyma, idioblast, latissifer and leaf epidermal cell, along with the extracellular space, and the cell surface, because what we will what I will tell that some of these final monomeric indole alkaloids are secreted out to the leaf surface. That is again a new discovery, and that plays important role in defending the leaves from predators. So, I will not name the alkaloid today. So, let us keep it for the next class. So, with this I end this class.